as epigenetic testing becomes more common, it's really important we clear up a common misunderstanding. And that's the confusion between your chronological age and something called your epigenetic age. So, what is epigenetic age? It's a biomarker. It's a number that's calculated by looking at the methylation patterns at hundreds of specific spots across your genome. And while it does tend to track with your chronological age, how many birthdays you've had, it is definitely not the same thing. What's actually more useful is a metric called epigenetic age acceleration. It's pretty simple. It's just the difference between your epigenetic age and your chronological age. If your epigenetic age is higher than your actual age, that's called positive age acceleration. And we know it's associated with a higher risk for disease and mortality. It's a dynamic measure of your biological aging process, not just the number of years you've been alive. All right, let's take a moment to recap the core ideas we've gone over. So let's review. First, epigenetics gives us a critical layer of control over our genes without changing the DNA sequence. Second, our diet, especially foods with methyl donors, provides the raw materials for this control through that key molecule, SAM. Third, the agouti mouse study showed us just how powerful these nutritional inputs can be, creating lasting, heritable changes. And finally, we can measure these patterns to calculate epigenetic age acceleration, which gives us a really powerful biomarker for biological aging. Okay, to finish up, let's run through a few questions to make sure these concepts stick. First one, what is the primary thing that determines a cell's ability to methylate its DNA? Question two, in that agouti mouse study, what was the specific molecular effect that the methyl donor diet had on the agouti gene? And for our last one, let's think about a real world application. How might compounds called polyphenols, which you find in things like green tea, influence the epigenome when it comes to cancer? And here are the answers. A cell's methylation potential really comes down to that SAM to SAH ratio. In the mouse study, the diet worked by increasing DNA methylation right at the gene's promoter, which silenced it. And as for polyphenols, they can actually inhibit the enzymes that add methyl groups, which could potentially reactivate tumor suppressor genes that cancer has silenced. This just goes to show how understanding these pathways is opening up entirely new avenues for both precision nutrition and medicine.